Welcome to our Section 4 video where we're going to discuss the endomembrane system and proteins. So our learning objectives are to list the components of the endomembrane system and recognize the relationship between the endomembrane system and its functions. So first, let's describe this system. It's basically a group of these different membranes and organelles in our eukaryotic cells, and they're going to work together to modify and package up and transport lipids and proteins. And it's going to consist of our nuclear envelope, our lysosomes, which we don't really have any pictured here, but they would kind of be hanging out over here. We also have vesicles, which we see here, our endoplasmic reticulum, this is our rough ER here, and our Golgi apparatus pictured here, and the plasma membrane, which we could see on the outside here. Now let's start off describing the endoplasmic reticulum. You could see that these are interconnected membranous sacs and tubules that are collectively going to modify proteins and synthesize or make lipids. These tubules are these hollow portions that we see, and we call them the lumen or cisternal space. Our membrane here is made up of a phospholipid bilayer, and you could see that they would be embedded with proteins, and we can also see that it is continuous right at this point with the nuclear envelope, and we may even have this part attached as well. Now we have two different types of endoplasmic reticulums. We have the rough endoplasmic reticulum, which we call RER, and that's because we have ribosomes that attach to the cytoplasmic surface. That's kind of what we saw over here where we have the ribosomes attached to the wall, and that gives it that rough appearance. And the ribosomes are also there because they transfer the proteins into the RER's lumen, where they're going to start to have structural changes take place, where we'll fold them and acquire side chains. So more molecules basically attaching to these proteins. And they can become incorporated into the membrane, and they can also be secreted from the cell as a protein hormone or an enzyme. So I'm going to go back to this image here so you can see that we would go through our RER, and then we're going to pass it, put it into a vesicle, and we could send it to other areas too in order to continue with some changes. They can also help make the phospholipids for the plasma membrane and put them into vesicles so that they can reach their destination. So as you can imagine, we would find a lot of RERs in cells that have a lot of proteins, for instance, like our liver cells. Now, the other type of ER that we have is smooth ER, and this is going to be continuous with the rough ER, which we see pictured here, but it's going to have few or no ribosomes on its cytoplasmic surface, so hence the name smooth. And its function is to synthesize carbs, lipids, and steroid hormones. We can also use it to detoxify medications and poisons and store some calcium ions. So in our muscle cell, we're going to have a specialized smooth ER, which is known as the sarcoplasmic reticulum. And this is where we are going to store all of the calcium for our muscle cell to use for contraction when we're ready. Next, we have our Golgi apparatus or Golgi body, and this is also going to be a series of flattened membranous sacs like we see here. The cis face is going to be closer to the ER. Okay, so I think I have another image of this just to show you. So here is our nucleus, the rough ER, and then this is the cis face. Notice how it's facing the rough ER, and then on this side we're seeing the trans face, which is facing the plasma membrane, and its job is to sort, package, and tag the proteins and also lipids that it's going to receive through the vesicles from the smooth ER and the rough ER. And these vesicles first go to the cyst face. They're going to fuse, empty those contents into the lumen where they'll undergo modifications. And commonly they'll add in some short sugar molecule chains and then tag it with a phosphate group or just smaller molecules. And then we'll package it into secretory vesicles and it'll leave through that trans phase. We will have an abundance of Golgi in areas like our salivary cells because they're secreting digestive enzymes, immune cells because they'll be secreting antibodies, and within plants, Golgi will synthesize polysaccharides, which can be incorporated into the cell wall. So here's another note on lysosomes, and I'm going to show you how they really function. And we said that our lysosome will have these digestive enzymes or hydrolytic enzymes so they can digest either waste or pathogens, meaning they're organisms that cause disease, like bacteria, for instance. So we have this special immune cell called a macrophage, which is a white blood cell, and it has the ability to go through this process called phagocytosis. Phago means to eat, and cyto, of course, is referring to the cell. So what it does is it will bring in the particle, whether it's food, waste, or a pathogen, and we will call this a food vacuole in this case, but it can come together and fuse with a lysosome so that we could get all of the digestive enzymes to then start breaking apart this food particle. Now, when we ingest a pathogen, we would call this a phagosome, and that phagosome fuses with the lysosome, and then we call it a phagolysosome. And we digest the contents, and then it will leave the cell through exocytosis. Whereas when we were bringing that into the cell, we call that endocytosis.